The total number of cancer cases, has been steadily increasing over the years, mainly because people are living longer today than they did in the past, and cancer risk increases dramatically with age. More than 100 types of cancer can be distinguished from one another based on differences in their site and origin and the cell type involved. Some cancers have been increasing or decreasing in frequency on the age adjusted bias, but the rates for most cancer have not changed much during the past century. Hypertro- Hypertrophy and hyperplasia are normal types of tissue growth based on an increase in cell size or number, respectively. Dysplasia is an abnormal type of cell proliferation involving the loss of normal cell and tissue organization. Although it is potentially reversible, dysplasia can also evolve into neoplasia. In neoplasia, cells proliferate in an uncontrolled, relatively autonomous fashion that produce tumors in which there is progressive increase in the number of dividing cells. Benign tumors grow in a confined local area and so are rarely dangerous, whereas malignant tumors are potentially life-threatening because they can spread to distant sites by invasion and metastasis. Malignant tumors are referred to as cancers. Survival rates differ significantly for various kinds of cancer, ranging from better than 99% 5-year survival for certain kinds of skin cancer to less than 20% 5-year survival for cancer of the lung, esophagus, liver, and pancreas. In tumor grading, cancer of the same type are assigned numerical grades based on differences in microscopic appearance. In tumor staging, cancer of the same types are assigned numerical stages based on tumor size and how much the tumor has invaded and metastasized at the time of initial diagnosis. The remainder of the text is organized around the following question: what kind of cellular abnormalities allow cancer to grow and spread in unrestrained fashion? What causes cancer? What role do genes play in development of cancer? How is cancer detected and treated? Can cancer be prevented? Key terms: hypertrophy. Tissue grows based on increase in size. Hypertrophy. Hyperplasia. Tissue grows based on increase in the number of cells, but cell organization is normal. Dysplasia. Abnormal tissue growth in which cell and tissue organization is disrupted. May be an early stage in cancer development. Neoplasia. Abnormal growth process in which cells proliferate in an uncontrolled, relatively autonomous fashion. Neoplasm. Growing mass of tissue created by an abnormal process in which cells proliferate in an uncontrolled, relatively autonomous fashion, leading to continual increase in the number of dividing cells. May be either benign or malignant or called a tumor. Cell differentiation. Process by which cells acquire the specialized properties that distinguish different types of cells from each other. Basal cell. Relatively differentiated type of epithelial cell whose proliferation gives rise to cells that are more specialized. Division of basal cell in the skin produce more basal cell plus cells that migrate toward the outer layer of the skin. gradually differentiating into flattened squamous cells. Squamous cells. Epithelial cell exhibiting a thin, flattened shape, found in the outer layer of the skin and in several other types of epithelial tissue. Benign tumor. A tumor that grows only locally, unable to invade neighboring tissue or spread to other parts of the body. Malignant, malignant tumor. A tumor that can invade neighboring tissue and spread through the bloodstream to other parts of the body, also called a cancer. Metastasis. Metastasis. Spread of tumor cell from one part of body to other via the bloodstream or other body fluid. Cancer. Uncontrolled growing mass of cell that is capable of invading neighboring tissue and spreading via body fluid, especially the bloodstream, to other parts of the body, also called a malignant tumor. Carcinoma. A malignant tumor arising from the epithelial cell that cover external and internal body surfaces. Epithelial cell. Type of cell that forms a covering layer over external and internal body surfaces. Sarcoma. Any cancer arising from a supporting tissue, such as bone, cartilage, fat, connective tissue, or muscle. Lymphoma. Cancer of blood or lymphatic origin that grows mainly as solid masses of tissue. Leukemia. Cancer of blood or lymphatic origin in which the cancer cell reside mainly in the bloodstream rather than growing as solid masses of tissue. Primary tumor. Tumor at the site where cancer initially arose. Five-year survival rates. Percentage of people who are still alive 5 years after diagnosis of a disease. Lead time bias. Tendency for survival rates to appear to be improved by early diagnosis. Polyp. Any mass of tissue that arises from the wall of hollow organ and protrude into the lumen. Biopsies. Cutting out a small sample of tissue for microscopic examination to determine whether cancer is present. Pathologist. Doctor who specialize in diagnosis disease by examining tissue and cells with a microscope. Mitotic index. Percentage of cells in a population that are undergoing mitosis at any given moment. Tumor grading. Assignment of numerical grades to tumor based on differences in their microscopic appearance. Higher grade cancer tend to grow and spread more aggressively and be less responsive to therapy than lower grade cancer. Anaplastic. Poorly differentiated and abnormal in appearance. Tumor staging. Estimate of how far cancer has progressed based on a tumor size and extent of spread at the time of diagnosis. Also see TNM system. TNM system for assessing tumor stage that consider three criteria: T for tumor size, N for lymph nodes involvement, and M for metastasis. Oncogenes. Oncogenes are genes whose presence can lead to cancer. Some oncogenes are introduced by virus, but in human cancer, most arise by mutation from normal cellular genes called proto-oncogenes. Nucleotide. Guanine, cytosine, adenine, thymine, complementary base pairing, gene transcription, uracil, messenger RNA, translation, codon. These are everything that I know. Chapter 2. Chapter 2. Summary of main concepts. Cancer cells exhibit a distinctive collection of abnormal traits, many related to uncontrolled cell proliferation and survival, that together constitute the profile of a typical cancer cell. Isolated cancer cells can be identified by their ability to form tumors when injected into immunologically deficient mice. Cancer cells grown in culture are anchorage independent, exhibit a decreased susceptibility to density dependent inhibition of growth, and proliferate indefinitely because they possess mechanism for replenishing telomere sequences. Cell proliferation is normally controlled by growth factors, which binds to cell surface receptor that triggers intracellular signaling pathway leading to cell growth and division. This signaling pathway acts in part of fostering the production and activation of CDK cycling complexes. One type of CDK cyclin phosphorates the RB protein, which control passes through the restriction point and into S phase. Will not normally divide unless they are stimulated by an appropriate growth factor, but this restraint is circumvented in cancer cells by various mechanisms that create a constant activation of growth, signaling the pathway even in the absence of growth factors. Checkpoint pathway monitor the cells for DNA damage. DNA replication and chromosome attachment to the spindle and transiently halt the cell cycle if conditions are not suitable for continuing. Cancer cells exhibit defects in checkpoint pathway, as well as loss of restriction point control. As a result, cancer cells continue proliferating under conditions in which the cell cycle of normal cells will stop. Apoptosis is a cell death pathway that is invoked for destroying unneeded or defective cells. During apoptosis, cells are destroyed by protein-degrading enzyme called caspases. The p53 protein plays a key role in triggering apoptosis in cells that has incurred DNA damage. In cancer cell, defects in p53 or other components of a apoptotic pathway allow the cell to evade apoptosis. Mutation play a central role in triggering the uncontrolled proliferation of cancer cell. Mutations are changes in the base sequence of DNA that arise spontaneously or in response to mutagenic chemicals or radiation. Translation synthesis of DNA and excision repair are used to correct mutation involving mismatches bases and non-homogeneous and joining and homogeneous recombination are used to repair double-strand DNA breaks. Cancer cells often exhibit defect in this DNA repair mechanism, allowing cells to accumulate mutation at rates that are significantly higher than normal. Cancer cells frequently exhibit gross chromosomal abnormalities, including deleted chromosome segments, chromosomal translocation, and the loss of some chromosome and extra copies of others. DNA isolated from the chromosomes of some human cancer cells has been shown to cause cancer when transferred into normal cell under laboratory conditions. Certain cancer exhibit tumor-specific antigens that are capable of eliciting an immune response. The immune surveillance theory postulates that such an immune response can protect people from developing cancers. Many cancer cells, however, have devised ways of evading immune attack. Other molecular changes detected in cancer cells include decreased production of protein involving in cell adhesion and gap junction formation, and an increased production of embryonic proteins, proteases, and proteins that stimulate blood vessel growth. Key terms: density dependent inhibition of growth, tendency of cell division to stop when cell growing in culture reach a high population density. Anchorage dependent trait exhibited by the cell of normal tissue which must be attached to a solid surface such as extracellular matrix before they can proliferate. Anchorage independent trait exhibited by cancer cells which grow well not just when they are attached to a solid surface but also when they are freely suspended in liquid or semi-solid medium. Extracellular matrix. Insoluble meshwork of protein fibers and polysaccharides that fill the spaces between neighboring cells. Anarchists. Cells that trigger by lack of contact with the extracellular matrix. Clone. Population of identical cells produced from a reproduction of single parental cell, also applied to gene or organism. Telomere. End of linear chromosomal DNA molecule contain the same short base sequence repeated over and over again. Telomerase. Enzyme that adds new copy of the telomeric repeat sequence to the ends of existing DNA molecules. Growth factors. Class of extracellular signaling proteins that stimulate, or in some cases inhibit, the proliferation and particular cell types by binding to specific receptor proteins located on the outer cell surface. Platelet-derived growth factor, PDGF, protein produced by blood platelets that stimulates the proliferation of connective tissue and smooth muscle cell. Epidermal growth factor, EGF, protein that stimulates the growth and division of a wide variety of epithelial cell types. Receptor. Protein that contains a binding site for a specific signaling molecule. Plasma membrane. Membrane composed of lipid and protein that defines the outer boundary of the cell and regulates the flow of materials into and out of the cell, also called the cell membrane. G1 phase, stage of cell cycle between the end of previous cell division and the onset of chromosomal DNA replication. S phase, stage of the cell cycle when the chromosomal DNA is replicated. G2 phase, stage of the cell cycle between the completion of chromosomal DNA replication and the onset of cell division. M phase, stage of the cell cycle when nucleus and the rest of the cell divides. Series of stage cell cycle, series of stages known as G1, S, G2, and M phase that are involved in preparing for and carrying out cell division. Mitosis, division of the nucleus during the M phase of the cell cycle. Cytokinesis, division of the cytoplasm during the M phase of the cell cycle. Interphase, portion of the cell situated between successive cell division M phase, composed of G1, S, and G2 phases. Restriction point. Control point near the end of G1 phase of the cell cycle, where the cycle can be halted until conditions are suitable for progression into S phase, regulated to a large extent by the presence or absence of extracellular growth factors. Cyclin dependent kinase, CDK. Any of several protein kinases that are activated by different cyclins and that control progression through the cell cycle by phosphorylating relating various target proteins. Protein kinase. Protein kinase, any of numerous enzymes that catalyze protein phosphorylation, that is, the addition of phosphate groups to protein molecules. Phosphorylation. Phosphorylation. Addition of phosphate group. Cyclin. Any of group of protein that activate the cyclin dependent kinases that are involving in regulating progression through the cell cycle. Protein phosphatase. Any of numerous enzymes that catalyze protein dephosphorylation, that is, the removal of phosphate group from protein molecules. RB protein. Protein whose phosphorylation controls passage through the restriction point of the cell cycle. Checkpoint. Pathway that pathway that monitors condition within the cell and transiently halt the cell cycle if conditions are not suitable for continuing. DNA replication checkpoint. Mechanism that monitors the state of DNA replication to ensure that DNA synthesis is completed prior to permitting the cell to exit from G2 and begin mitosis. Spindle checkpoint. Mechanism that halt mitosis at the junction between the metaphase and anaphase if chromosomes are not properly attached to the spindle. DNA damage checkpoint. Mechanism that monitors for DNA damage and halt the cell cycle at various points, including late G1, S, and late G2, if damage is detected. P53 protein. Molecule that accumulates in the presence of damaged DNA and activate genes whose products halt the gene cycle and trigger apoptosis. Apoptosis. Cells cells are mediated by a group of protein degrading enzyme called caspases. Involves a programmed series of events that leads to the dismantling of the internal contents of the cell. Apoptosis bodies. Cell fragments produced as a cell is dismantled by the process of apoptosis. Caspase. Any of a family of proteases that degrade other cellular proteins as part of the process of apoptosis. Mutation. Changes in the base sequence of DNA molecules. Double helix, complementary base pairing, mutagen. Chemical or physical agent that is capable of including mutation. Pyrimidine dimer. Pyrimidine dimer. Mutation in which two adjacent pyrimidine bases, CC, CT, TC, or TT, in DNA are joined together by a covalent bond. Pyrimidine dimer distort DNA structure, thereby causing incorrect nucleotides to be inserted during DNA replication. DNA polymerase. Any of group of enzyme involved in DNA replication and repair that catalyze the addition of successive nucleotide in the 3' end of growing DNA strand, using an existing DNA strand as a template. Translation synthesis. DNA replication across region where the DNA template is damaged. Excision repair. DNA repair mechanism that removes and replaces abnormal nucleotides. Mismatch repair. DNA repair mechanism that detects and corrects base pairs that are improperly hydrogen bonded. Non-homologous end joining. Mechanism for repairing double-strand DNA breaks that use protein that are bind to the end of the two broken DNA fragments and join them together. Homologous recombination. Method of fixing double-strand break in DNA, which an intact copy of chromosomal DNA molecule serve as a template for guarding the repair of the broken DNA, also used for the exchange of genetic information between the DNA molecule exhibiting extensive sequence similarities. Genetic instability. Trait of cancer cell in which abnormally high mutation rates are caused by defects in DNA repair or chromosome sorting mechanism. Diploid, containing two sets of chromosome and therefore two copies of each gene, can describe a cell, nucleus, or organism composed of such cells. Aneuploid, containing an abnormal number of chromosomes. Philadelphia chromosome. Fragment of chromosome 22 found in the cancer cell of about 90% of people with chronic myelogenous leukemia generated by a reciprocal exchange of DNA between chromosome 9 and 22. Transfection. Transfection. Transfer of foreign DNA into cells under artificial condition. Antigen. Any foreign or abnormal substance capable of triggering an immune response. Antigen presenting cell. Cells that degrades and process antigen and displays the resulting antigen fragment on its surface to activate an immune response by lymphocytes. Major histocompatibility complex, MHC, type of cell surface protein recognized by the immune system as part of the process of including an immune response. Lymphocyte. Class of white blood cell involve immune responses. B lymphocytes produce antibodies, whereas cytotoxic T lymphocytes kill targeted cells directly. B lymphocyte. Class of immune cells specialized for producing antibodies. Cytotoxic T cytotoxic 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 T lymphocyte. Class of immune cells specialized for detecting foreign or abnormal cells or cells that have been infected with an invading bacterium or viruses. Immune surveillance theory. Theory postulating that immune rejection of cancer cells help protect people against the development of cancer. Lectin. Carbohydrate binding protein possessing two or more carbohydrate binding sites, thereby allowing a single lectin molecule to link two cells together by binding to carbohydrate groups exposed on the surface of each cell. Gap junction. Structure involved in cell-to-cell communication that joins cells together in a way that allows small molecules to pass directly from one cell to another, constructed from a protein called connexin. Chapter 3. Summary. And angiogenesis is required before tumor can grow beyond a few milliliters in diameters. Control over angiogenesis is exerted by a finely tuned balance between activator protein, such as VEGF, and FGF, and inhibitor proteins such as endostatin, angiostatin, and thrombospondin. VEGF and FGF produced by tumor cells activate angiogenesis by binding to receptor present on the surface of endothelial cells, thereby stimulating the cells to proliferate. produce matrix metalloproteinase, migrate into surrounding tissue, and assemble into new vessels. Treating animals with angiogenesis inhibitors can inhibit the growth of primary tumors as well as formation of metastasis. After angiogenesis has occurred, cancer cells evade surrounding tissue, enter the circulatory system, and metastasize to distant sites. Each of these steps involve numerous interactions between cancer cells and normal cells of the host. The ability to invade neighboring tissue and penetrate vessels walls is facilitated by three properties exhibited by cancer cells. Decreased cell-to-cell adhesion, often traced to a loss of E-cadherin, increased motility, and secretion of proteases that facilitate degradation of the extracellular matrix and basal lamina. Tumor consists of heterogeneous population of cells that vary in the ability to metastasize, their susceptibility to immune attack, and the site of which they preferentially metastasize. Only a tiny fraction of the cancer cells that enter the circulatory system survive the trip and establish successful metastasis. Cancer cells tend to preferentially metastasize on the organ containing the first capillary bed encountered after entering the bloodstream. For this reason, cancer arising in the gastrointestinal tracts often metastasize into liver, and most other cancer frequently metastasize to the lung. Many cancers also exhibit an organ-specific pattern of spread, preferentially metastasizing to particular sites that provide a congenial environment for their growth. Cancer is a disease whose properties change with time as tumor progression ill cells with increased growth rate, invasiveness, ability to survive in the bloodstream and evade immune attack, ability to grow in other organs, and resistant to drugs used in cancer treatment. The study of metastasis promoting and metastasis suppressor gene is helping scientists identify potential targets for the development of new drugs designed to halt cancer spread. Keywords: angiogenesis, angiogenesis, growth of new blood vessels. Endothelial cell, type of cell that lines the internal surface of blood and lymphatic vessel. Tumor angiogenesis, process by which cancer cells stimulates the development of a blood supply. Vascular endothelial growth factors, signaling proteins that plays a central role in stimulating the growth of new blood vessels. Matrix metalloproteinases, family of enzymes that degrade protein found in extracellular matrix, referred to as a metalloproteinases because they require zinc or calcium atoms to function properly. Angiostatin, protein that inhibits the growth of blood vessels, a fragment of the protein plasminogen. Endostatin, protein that inhibits the growth of blood vessels, a fragment of the protein collagen. Thrombospondin, signaling protein that inhibits the growth of blood vessels. Tumor dormancy, formation of microscopic tumor masses that remain dormant for a prolonged period of time. Metastasis. Metastasis. Spread of tumor cell from one part of the body to another via the bloodstream or other body fluid. Cell-to-cell adhesion protein. Class of protein located at the outer cell surface that cause cells to adhere to one another. Cadherin. Cell-to-cell adhesion protein that binds epithelial cells to one another. Protease. Class of enzyme that degrade protein molecules. Plasminogen activator. Protease that converts plasminogen into plasmin. Plasmin. Protease produced from precursor of plasminogen by action of plasminogen activator. Tumor progression. Gradual changes in tumor property observed over time as cancer cells acquire more aberrant trait and become increasingly aggressive. Major histocompatibility complex. Type of cell surface protein recognize the immune system as part of process of inducing an immune response. Cytotoxic T lymphocyte. Class of immune cells specialized for attacking foreign or abnormal cells, or cells that have been infected with invading bacterium or viruses. Tumor host interaction. Interaction between tumor cells and surrounding normal cells that influence tumor cell proliferation, invasion, and metastasis. Metastasis promoting genes. Class of gene coding for proteins that are required for active events associated with invasion and metastasis. Metastasis suppressor gene. Class of gene coding for protein that inhibits or block events associated with invasion and metastasis.